Progress in chemoprevention drug development, the promise of molecular biomarkers for prevention of intraepithelial neoplasia and cancer, a plan to move forward. The development of effective chemoprevention drugs for intraepithelial neoplasia and cancer remains a challenge. Molecular biomarkers have the potential to aid in personalized approaches to chemoprevention. Biomarkers can increase the efficiency and accuracy of drug development by identifying high-risk populations and monitoring drug efficacy. Molecular biomarkers can provide insight into the mechanisms of carcinogenesis and identify potential targets for chemoprevention. Biomarkers can be used to develop personalized approaches to chemoprevention and monitor drug efficacy. Biomarkers can also aid in the selection of high-risk populations for clinical trials. The identification of effective chemoprevention drugs remains a challenge due to the complexity of carcinogenesis and the diversity of cancer types. Clinical trials for chemoprevention drugs often require large sample sizes and long follow-up periods, which can be expensive and time-consuming. The development of innovative clinical trial designs and biomarker-driven approaches can increase the efficiency and accuracy of drug development. Chemoprevention drug development can involve the identification of new molecular targets and the development of drugs that target multiple pathways. Combination therapies may increase the efficacy of chemoprevention drugs. Biomarker-driven clinical trial designs and innovative statistical methods can improve the efficiency and accuracy of drug development. Molecular biomarkers can aid in the early detection of cancer by identifying precursor lesions and monitoring disease progression. Biomarkers can also be used for risk assessment and to identify high-risk populations for screening and chemoprevention interventions. Advances in imaging technologies can further improve the accuracy and sensitivity of biomarker-based approaches to early detection and risk assessment. Mechanisms of action. Chemoprevention drugs can target multiple pathways to prevent cancer. Combination therapies may increase the efficacy of chemoprevention drugs. Continued research in identifying new molecular targets is essential for the successful development of effective drugs. Clinical trials. Biomarker-driven clinical trial designs can increase the efficiency and accuracy of drug development. Innovative statistical methods and imaging technologies can further improve the accuracy of drug development. Integration of these methods with clinical trial designs can help in early detection and prevention of cancer. Challenges. The development of chemoprevention drugs is challenging due to the complex nature of cancer. Toxicity and adverse effects are important considerations in drug development. The cost of drug development is a major challenge for bringing drugs to market. Opportunities. Developing effective chemoprevention drugs has the potential to significantly reduce the burden of cancer on society. Molecular biomarkers offer opportunities for personalized approaches to chemoprevention. Continued research and investment are necessary to overcome the challenges and seize the opportunities in this field. Call to action. The medical community must prioritize the development of effective chemoprevention drugs. The use of molecular biomarkers, personalized approaches, and innovative clinical trial designs can significantly improve drug development. The development of effective chemoprevention drugs can reduce the burden of cancer, making it an important area of focus for continued research and investment.